Hi everybody. So the summer is over, we are approaching early autumn, and you are again at the channel My-Genome-Enhancer. Today we will analyze data published in 2020 on ovarian cancer cell line which was treated by an anti-cancer drug paclitaxel to develop resistant cell lines. These cell lines were subject for RNA-seq analysis, which then were analyzed by us using Genome-Enhancer, and we revealed several mechanisms how these cancer cell lines were developing resistance to that drug. Finally, the Genome-Enhancer predicted several drugs which potentially can be used in combination with paclitaxel to overcome that resistance, depending on which molecular targets and which biomarkers are expressed in that cell line. The software can predict which drug can be more effective. And finally, we can validate those findings of the potential combinations of these drugs with paclitaxel in the literature, where in recent years these combinations were subject of various studies confirming their high potential to overcome resistance, and some of these combinations were even leading to several clinical trials. Some of them confirmed that these combinations indeed are effective. Last time we were analyzing gene expression data and genomic data for one particular cell line of lung cancer, and we found out that this lung cancer cell line is sensitive to a certain drug, paclitaxel, which you can see here on this screen, the entry from Wikipedia. By the way, the link to that video is somewhere here, over there. So please have a look. You will see how we analyzed this data and how we found drug targets and the whole mechanism. But we know that paclitaxel, although it's a well-known drug, is used in many cancers, and it's used as a chemotherapy, but there are many cases when the cancer develops resistance to that drug. And we got interested to understand the resistance mechanisms and to find potentially drug targets, additional drug targets, which can be used to overcome that resistance, and maybe even some drugs which can be used in combination with paclitaxel. So, and to do that, we look at this paper, which was published in 2020 by these authors, and they studied a transcriptome of certain cell lines. They actually took a cell line, of ovarian cancer cell line, and they, using the paclitaxel with different increasing concentration, they developed resistant cell line. They actually developed several resistant cell lines under different concentration of paclitaxel. And they made a transcriptomics analysis, so they made actually RNA-seq, and they published this data in this GO entry. So basically we can look at this entry, and what we can see here, they stored the data of RNA-seq for the control cell line, so the initial cell line which is sensitive to that drug, and they also they stored this RNA-seq data for all the developed cell lines, where they used actually either small concentration of paclitaxel, like 4 nanomolar, or until the 128, very high concentration of that drug. But still they were able to create this resistant cell line. And now we can get this data and put it in our tools and analyze it. Let's watch it how we do it. We log in into the system, into the Genome-Enhancer. Here we are, then we create a project, let's call it ovarian cancer resistance to paclitaxel. Now we can load the data, so we go there to this gene expression omnibus entry. As you can see, we have this data here, and here at the end of the entry we can copy the link. And now we can put it into this system. So it will be automatically loading it directly from that server. Here we specify the data type, transcriptomics. Now it's importing into the Genome-Enhancer. We can go next. Now we have this data inside. Basically, this is a table with all the read counts for all the samples. The first two lines are the control and then each cell line. Now we can put this table into this gray box, so we can select these entries. And okay, first we have to put two duplicates for the control, and we'll have first for the smallest concentration of PTX, we have this entry here for PTX, and then we will create now several boxes for all other conditions. So let's have a look. So for all conditions like 4 nanomolar, 16, 32, 64, and 128. And we'll put this data inside of each of these boxes. Now they are in, so we can go next step. And here we have to select the disease type, and basically this is ovarian neoplasm. And now we have to select all these conditions. So first we put the control, and then again 4, 8, 16, 32, 64, and 128. And we set control as a baseline. And we start the analysis. So as a previous time, the analysis will take several hours. While Genome-Enhancer is running, I will take a little walk. Results are ready, so let's have a look. So we see the full report, and the program found these two genes as the most important promising druggable targets in the context of this resistant to paclitaxel. And we can have a look at the whole report. It's a bit complicated. It's more than it was before, you see, because there are several categories which we're comparing to each other. So each of the category was compared to the control, and the program revealed upregulated genes and downregulated genes. You can see them here. So for instance, upregulated genes upon 16 PTX versus control, downregulated genes 16 PTX versus control, and so on. So all of the comparisons were done automatically. And then the program calculated this heat map, which puts each of the conditions here, and selected actually signal transduction genes because they are most important for our further analysis. And as you can see, the first two columns correspond to the control, but then you see that those cell lines are so a bit different to each other. Although all of them are resistant to the paclitaxel, but they probably developed this resistance in a different way. So they created different mechanisms of resistance. And you can see in some of the cell lines which were developed, certain genes are upregulated, and in other cell lines, the same genes are downregulated. But you can see that many of those genes involved in signal transduction pathways, they're playing a role here. In some cell lines, they are upregulated. In some cell lines, they are downregulated. And then each of the comparison and each of the set of the genes, upregulated genes, for instance, in the 16 PTX versus control, these 1,530 significantly upregulated genes were then mapped to the gene ontology. And as you can see, it revealed several important gene ontology terms. We combined several results from these comparisons, from these 6 comparisons, and put them on the same diagram. So you can see these diagrams, 2 diagrams, and as you can see, these are 4 PTX, so the 4 nanomolar, 8, 16, 32, 64, 128. And on this axis, we put the logarithm of p-value of the particular gene ontology term, which is found to be enriched in that particular comparison. And as you can see, several gene ontology terms, they are getting very important on the small concentration of that drug. And then, for instance, we see that this one is a regulation of neuron differentiation. So basically, the cell lines start using various cell differentiation mechanisms to develop resistance to that drug. Or, for example, the mesenchymal cell development is also getting important under the small concentration of the drug and less important under the higher concentration. Various such terms as the regulation of cell communication is important under all concentration. And another side, actually, several geocategories which are not very important under a small concentration of the drug, they're becoming important when the concentration of the drug gets very high. And as you can see here, for instance, extracellular matrix disassembly, this mechanism getting very important for resistance. And for instance, the regulation of NF-kappa B signaling or cellular metabolic processes. So the cells start using metabolic processes, various metabolic processes to develop resistance. And now when the system look for the composite modules in the promoters of the upregulated and downregulated genes, we can see that, for instance, here in 128 PTX in comparison versus control, in promoters of downregulated genes, for instance, the program found this composite module, which is pretty good discriminating these promoters from the promoters of the non-changed genes. And among the transcription factors which it found here, you can see rel factors actually from the NF-kappa B pathway. You see FOX and SOX transcription factors and E2F, which is involved in cell cycle control. So many transcription factors which are actually fitting to that to those gene ontology categories which we found in the first place. Next, Genome-Enhancer reconstructed the networks, the regulatory networks, which potentially are involved in regulation of genes in those cell lines. So, for instance, this network is built for the comparison 128 PTX, so very high concentration of the drug. And this network, as you can see, here has upregulated genes, these blue boxes, and on top of it, so upstream from those genes, we see those master regulators which are predicted to be potentially involved in regulation of those genes. We can click here to see the full diagram. We can zoom in and zoom out here to see more details of that diagram. Let's have a look at the master regulators. Here you see also the fold change. This color shows the fold change of those molecules observed in different comparisons. So we have 6 comparisons, and we have respectively 6 stripes here. And then some of the molecules like MEC1, you see it's upregulated in all 6 comparisons and all 6 cell lines resistant to that drug. But for instance, this molecule is under small concentration of the drug. This molecule is downregulated, but upon the high concentration, it's upregulated. So, and you see here that some of the master regulators like pol-like kinase 2 or EGF complex with EGF receptors, they are involved here in regulation of those genes, and they become the most important potential drug targets for us. Finally, Genome-Enhancer integrated all found master regulators into one schema, unified schema. So combining several master regulators which are found in different comparisons into such schema, we can see that we always see their integrins are playing important roles, various components of proteasome, and some other important molecules in this signal transduction network. At the end, Genome-Enhancer used these predicted drug targets to compute a list of drugs which are connected to those drug targets and which are FDA-approved drugs or used in clinical trials against ovarian cancer. and it computed and ranked those drugs according to their potential to be used to overcome resistance against paclitaxel. And at the top of this list you see pazopanib, then sorafenib, and the third one, dasatinib. And we are going to look at the literature to see if those combinations with paclitaxel of these three drugs are actually used, and what are the results of those combined use of these drugs. As we can see from the literature search, the combination of pazopanib and paclitaxel is actually used in several clinical studies as potentially beneficial. For instance, in this early paper in 2016, they made a phase 2 study of this combination against refractory urothelial cancer. And they actually shown, and we look at the conclusion, that they got the very promising results, and they found that 54% of the patients they benefit from that combination. But unfortunately, in another study, when this combination of pazopanib and paclitaxel was used against ovarian cancer, as initially in the study we analyzed, this clinical trial actually failed. And the conclusion of that clinical trial is that adding pazopanib and paclitaxel didn't improve efficacy, but instead increased toxicity and compromised chemotherapy delivery. So as you can see that although the primary results can be very promising, or in certain cancer it can work, or if for a certain patient it can work, generally in clinical trial, in real world, these combinations may not work. Again, when we look for another drugs from our list, for instance, paclitaxel and sorafenib, the very recent papers showing that actually this combination indeed works very well, especially on cell lines, and this is effective combination. And actually, as you can see here, they show that it's suppressing self-renewal of cancer stem cell. And we show also in our results the property of stem cellness are affected in the data we analyzed. But indeed, and this combination is indeed very promising. But again, when certain clinical trials were done for the combination of paclitaxel and sorafenib, for instance, for advanced renal cell carcinoma and breast cancer. So the results initially promising, and they found this combination works, but in the randomized trial of paclitaxel and sorafenib in HER2-negative metastatic breast cancer, the conclusion was that this combination wasn't found to be superior to paclitaxel monotherapy. Again, as we can see, this the real world is much more complicated than what we can see on cell lines. We have to observe the side effects of the therapy, and by combining several therapies, side effects may either decrease or increase. In most cases, it's increasing. So that's why real clinical studies are necessary. As for the third drug, dasatinib, again, the dasatinib and paclitaxel was used in combinations, and several studies indicated that dasatinib can enhance, for instance, paclitaxel inhibitory effect in pancreatic cancer cells, and that's done on cell lines. What we see from the first clinical trials of dasatinib and paclitaxel, for instance, for recurrent endometrial cancer, we see here the positive effects that this clinical trial shows a little bit of positive effect of this combination. So these all studies shows to us that what we find is potential good combination of the therapies has to be checked individually. So in big clinical trials, when many individuals are used, these combinations often may not work. But in each individual patient, it may work, and we have to look at the additional biomarkers which can indicate the sensitivity or resistance now to that combination. And our studies and revealing of the mechanism of the resistance can help us to find additional molecular biomarkers which can determine or can show whether the particular combination of drugs can be beneficial for that sample and for that particular tumor observed in that patient. Thank you very much for your attention, and see you next time.